post-lunch sleepy feeling going on here. Thanks so much for doing all the hard work and organizing, Mika, and I just get to roll in. Haven't done anything really. And I want to thank Valerie for prompting for her comment, because a lot of what I want to talk about here is stuff that I have thought for a while I could do or maybe should do, but just never was really sufficiently motivated to do. But then there was this talk, and I was like, well, maybe now's the time to do it. So when she said, you know, we need moments, we need these sort of events to motivate people to actually finish things, it's true. Case in point. And so today what I want to do is actually, it's a little bit more, less of a, I've already done all of this stuff kind of talk. It's a little bit more of a hopeful pitch about what we might be able to do kind of going forward, dealing with all this scaling up stuff and climate change. So to get myself in the right frame of mind, I went back to the G2P2POP website and just refreshed my memory on the goal, make sure I'm kind of hitting the goal correctly. You already heard this, but predicting how populations and species of vertebrates will respond to a changing climate by integrating knowledge across levels of biological organization. It's a very good goal, right, but a very big goal, right? And even inside of that, right, there are things that we might consider to be Herculean tasks on their own, right? Like predicting stuff, right? Like that's not easy, right? We don't know how to do that. We sort of know how to do that a little bit. Climate change, well, that's not easy. You know, it's really quite a mix of phenomena, not to mention where does it actually impact things. Cross levels of organization, we also know that that's also a big task, right? Most of us are trained within levels, right? We learn how to work on specific things at specific levels with specific tools and systems. Moving between them is actually a considerable challenge. So nested in this big goal are many big goals. But I'm going to try to touch on them a little bit in my typical way, trying to simplify everything. And so unlike a normal seminar where you might have conclusions, right, you know, today I'm going to have suggestions, little points of advocacy, if you will, right, instead of conclusions. And these are them, right? When we're making models, we should try to anchor them, and crossing levels, that is, try to anchor them at places that mathematically must be true. And I'm going to give you an example of that. To visualize parameters in our models as emergent properties of things that occur at lower levels of organization. Right? And then we take those parameters apart. Don't accept them as, you know, fixed things. They are generally functions that can be broken up. 3, to allow for changes in allele frequencies through evolution. Right? So if we are trying to predict from alleles to populations, and the alleles change, we actually have to account for that if the alleles matter. And then finally, a little promotional bit here, try out Gillespie Eco-evolutionary models, or GEMS, as a tool. It's a way you can do all of these things. And so I'm going to give you some examples of that. All right, so what's the landscape that we're talking about here? I had to put this slide together for myself, right, to think about this, right? We have levels of organization, or blues, if you will. Standard sort of genomes, the populations, sort of categories that we use to describe where things occur. And I think it's important to distinguish the name or the category from the states that we measure at those categories, right? So whereas we have like a category of populations, there are things at the population level that we measure, states, and there are many different states, right? So we might measure abundance at the population level, but you might also measure stability of a population, or the probability that a population goes extinct, or other kinds of age structure, or who knows what. There are many different kinds of states, and that goes for all of the other levels. When we say phenotype, right, that is shorthand for it's too big for me to say anything else, right? Phenotypes are everything, right? You know, so what are we measuring there? Different kinds of states, mass, lengths, you know, who knows what, right? At the cells level, you know, phenotypes or states at the cell level, they can be lots of things. This is already beyond my expertise, but I threw up networks here, like network structure as a possible thing that you might measure at the cell level. Molecular level, we have proteins, and of course genome level we have alleles, but it could also be gene copy number, right, other kinds of states. And when we're scaling up, right, there's, you know, remembering that we have these states at different levels, and we're scaling up, we need to be connecting the states, right? A measurement at one level and a measurement at another level. And there are essentially two ways of going about this. One is correlational, right? I'm simply going to assess the relationship between a state measured here and a state measured there, right? Statistical relationships. And this could include like allometric scaling things. I know the phenotype of something, and I know maybe the abundance of that thing. I can approximate the abundance of a 100-gram mammal, right, through a statistical relationship. Or, you know, gene to protein mass, right, statistical relationships between the presence of certain alleles and certain kinds of outcomes. The second way is to actually try to get at the mechanisms. And what we're thinking about there are processes that transform one state into another state, right? So there is a mechanism of gene expression that transforms an allele into something else that you can measure at another level, so a protein. And there are mechanisms that you could invoke at all of these levels of organization. And so this is where kind of, you know, I think in an ideal world, we would want mechanistic transformations across levels everywhere. But I think in the real world, we're not going to get that. We're going to have, we're going to substitute statistical relationships between states where necessary or where most convenient. All right, so one of the things, one of my suggestions is to be mindful of evolution here, right? And that's because evolution can be a lot faster than we tend to think of it as ecologists anyway, right? And the reason why it can be fast and something that you need to deal with is that it's essentially driven by the same process that drives ecology in the first place, right? Population dynamics arise through differences in birth and death rates, demography. Evolution arises through demography just because individuals differ in birth and death rates. The same very things that are causing populations to grow and decline are causing differences in fitness among individuals at the same exact time. So there should generally be feedbacks, right, from our phenotypes back to alleles. And I mean fast, right? Like, you know, the Darwin's finch example, right? There was a massive shifts in beaks in 1 year. And that's all coming from differences in phenotypes. The same thing that drove the change in population drove the shifts in the beak sizes. And if you take a strictly Darwinian view, right, a lot of this difference in phenotypes are coming from feedbacks within the population. So as the population grows and resources become limited, there are differences in individuals. Individuals with certain phenotypes can have more babies or survive better. And so that, there's a feedback then from abundance to your phenotypes. But I'm going to make everything worse, sorry, because as a community evolutionary ecologist, right, I know and I've observed that one of the biggest drivers of fitness differences among individuals are other species. The biotic world, predators, parasites, diseases, competitors. So really, if we want to do this thing, we probably do need to do this. Add one more level, sorry. Another level of organization, more states, more mechanisms, species interactions. Lauren's shaking his head. Right, but Valerie also prompted, I didn't prompt this, but gave me cover by talking about her trout. Right, she couldn't predict the outcomes of a stressor on her green cutthroat trout without considering the brown cutthroat trout competitor. Right, so, and there are no species that live alone out there. Right, so I think the community is essential.
But can I just say something? Can I add something here?
Yeah.
Is that thinking, and I'm seeing Lauren getting nervous, I think you can handle those external factors in a population model without explicitly adding another level of complexity, right? Because you treat them as parts of the environment, but your focus is still on your population. I mean, I think it's, you know what I mean?
Yeah.
You don't have to explicitly model multiple other species necessarily.
Yeah, but you might.
But you might want it. You might want it. Yep.
And abundance at the time. Right. They're evolving and responding to the climate at the same time.
True.
Right.
True.
I actually think that these interactions, these are the easiest. Right. So they're probably not that big of an This stuff, whoa. down where you can, right? And this may seem kind of simplistic, but there are mathematically places where you can write things down that must be true. And I think this mechanism here, demography, is one of those places, right? Populations only grow if there are more births than deaths, right? If you go from 10 to 11 individuals, you had a net 1 individual, right? It's really easy to write that down mathematically in a way that must be true. If you can anchor your models down in sort of mass balance, right, conservation law places, right, you can constrain everything else so that when you're connecting and adding models and putting pieces together, you don't sort of bust, right, the second law of thermodynamics. And it turns out if you do that here, it's pretty easy to start knocking things, to start working your way back down. All right, so I'm going to give you an example of this, very simple example, very cartoonish. It's going to have some math. I did notice that nobody else is throwing equations up. Sorry, I'm going to do it. I'm going to start here by talking about the logistic model. You guys all have heard about the logistic model, hopefully, right? Standard ecology. Logistic model is a modification of the exponential growth model, which is what I've got up there. And just to give you a quick reminder, DRDT is instantaneous rate of change in our population size R. Right? R max is some sort of maximum intrinsic rate of growth, and R is our state variable, our abundance. Right? And in the traditional exponential, right, this describes an accelerating population growth because R keeps getting bigger, right? So DRDT keeps getting bigger through time. Right? But that's if R max is a constant. If R is not a constant and it's just a thing, right? This equation is always true. Right? R is a function. This equation is always true. The rate of change in your population is always the instantaneous per capita rate times R. No matter what. It's just we don't know what R is. Like we don't know what makes up R. So this is a really nice place to start and say mathematically this must always be true. Now let me ask what goes into R and start breaking it down, dissecting it and going down levels. And that's what the logistic does. Now the logistic looks like this. It's basically in brackets R. Now it's not necessarily true that logistic is always correct, right? But it's always true that DRDT is equal to R. Right? But what this is going to do is going to allow, what I want to do here is break this apart. So what the logistic does is it says the growth rate of my population, the realized R, is really high when population abundance is low. And eventually it goes to 0 when the population abundance R is equal to K, because this term goes to 0 and my R goes to 0. But Rmax is a parameter. It's a population level observation, right? It's not an individual level thing. It's population level observation. And it can be dissected. It is an outcome of other things that are happening at lower levels. Right? I was going to change that. It's a little funky looking right now. I'm going to dissect it down into the next level of parts, right? And this is something that I call the BD logistic or the birth-death logistic. Rmax is got to be essentially the difference between the maximum rate of births and the minimum rate of deaths, right? So what we can do is we can break Rmax into two other parameters, and that's what I'm going to do here. Each one of those parameters now, births and deaths, can be a function of abundance in this way. So Bmax is really high when population abundance is low, and it goes down as the population grows. Bmin is really low when the population is small, and it goes up. And someplace they cross, and where births is equal to deaths, you have your carrying capacity. So we can write that out like this, where we simply have a birth function now and a death function, but they're all still in the brackets there, just R. Right, so whether or not those particular curves are true, I don't know. But if you see where I'm going, I'm trying to dissect my parameter, right, from the population level down. Right, so I've taken R, made it Bmax minus Dmin. And I've also gotten rid of K in this model because you can rewrite it. K is actually the function of 4 other parameters describing the relationship between abundance and births and deaths. All right, and these themselves are outcomes of lower levels, right? So the maximum rate of birth. What is that? Anybody? Break Bmax apart for me. How would you? What are some traits at the individual level that give me Bmax?
Quite clutch size or fecundity?
Clutch size, good one.
Gestational time?
Gestational time, like 1 over the gestational time, or that time between reproductive events times clutch size is Bmax, maximum clutch size, right? Okay, that's getting pretty close to individual level traits, right? Life history traits. Okay, clutch size, break that one out for me. Where does that come from?
Give us a hint.
Taxonomy. I mean, it's genetic.
Okay, so what is clutch size is just, do you have an allele for clutch size? That's my question. Or is there an allele for something that leads then to clutch size?
It may depend on environmental conditions, so there may be a maximum possible clutch size, but
Mhm.
there's a response variable.
So there might be phenotypic plasticity. It might actually be that there's some allocation rule that depends on the environment, and the allocation rule itself is there's an allele for that. Or maybe, you keep going. Anyway, you see what I'm doing here is you start from someplace that has to be true, and you keep dissecting. Eventually, you are going to get to something that you can connect to an allele. Right? That is possible. Here, all I've done is go to Bmax. Right? Well, so next thing I want to do, my other suggestion is to allow for evolution and to try out gems. So I'm going to have to give you a quick rundown on how these things work so that we can do it, and I'll show you what's going to happen. All right. So the Gillespie eco-evolutionary model is built on the Gillespie algorithm. Jonathan actually had the word up on one of his slides. It goes back to chemistry in the 70s. It's a way of simulating an ordinary differential equation, and it incorporates sort of a natural form of demographic stochasticity. And the way it works is you start with some model, say your DNDT model, you got some terms, or birth terms, death terms, some sort of rates of change. And when you solve an ODE normally, right, you're basically solving it as rates. This rate is adding to my overall rate, and this rate is taking it away. Right? In a Gillespie algorithm, you transform your rates into events. Right? So instead of having a rate of births, I have a birth event. And the probability of my birth event is given by my birth term, let's say it's B, divided by the sum of all the other terms. Right? So if you have a really big term, right, you have a high probability of an event like that happening. And what you do is you make a little pie out of it. And actually, we don't make a pie out of it, but visualize it as pie, right, where the size of each slice of the pie reflects the likelihood of an outcome. So in this case, our C term is really likely to happen. And because it's minus C something, this is like a death term, right? And when you run a Gillespie algorithm, you essentially spin this pie like a wheel of fortune, right? Right? And it's going to land somewhere, right? And it's most likely to land on the thing with the largest slice. That's the most likely thing to happen. But it could land anywhere, right? So if you do spin it and you land on a C, what you do is you take your population size 10 and you minus 1. That's it. You advance time, you go back, but all these things are functions of n, so then we have to recalculate our probabilities again, make pi, spin wheel of fortune. Let's say you get a birth right here, you add 1. And the population starts to go up and down this way, right? And if it's a population model, right, what you're doing is you're incorporating very naturally, automatically, demographic stochasticity, which is a very important part of population dynamics, right? Random variation in the sequence of events that are happening, sequences of births and deaths. This is how small populations go extinct, right? Through random chance of having extra death events more than you might expect. So you go through time, and you just keep running this thing until you decide to stop it. Right? If you run simulations over and over and over again, and you take the mean of it, it looks almost identical to the ODE solver, except you get all this noise around it that is accounting for demographic stochasticity. It's a very easy thing to code up to. A gem hijacks this original architecture and just inserts a step. Basically, we have all the same parts, but in a normal Gillespie algorithm, we take a parameter as a constant, right? So birth rate is 2, whatever, right? In a gem, instead, we initialize the population with a distribution of birth rates, right? And so this gets at individual variation. And instead of just saying, okay, my population is 10, giving it a number 10, I make a vector of, say, some trait that I'm interested in, let's say births here, right? A vector of growth with some sort of variance in it. And instead of using the mean birth parameter, I just choose one from my distribution. And then I calculate my pi slices from that and run back through. And if I happen to have a death event, I'm going to take that individual out. And if I happen to have a birth event, I'm going to add an individual that it looks a little bit like the parent, given some sort of underlying rule. And what we tend to use is a quantitative genetics rule of neuroscience heritability. Right? And so if you keep running this thing as a gem, what happens is your distribution of traits, which is what you have in a population, and when distributions of traits that are genetically controlled move, that's what we call evolution. Right? So if you have a trait that's tending to cause you to die, the distribution moves away from that trait towards one that causes you to live. And if you have a trait that tends to cause you to have lots of babies, your distribution moves in that direction. And so what you get is a supernatural computational analogy to natural selection. That's what happens in the world. You have a trait, it influences your demography, your fitness, chance of living, chance of reproducing, and if it's good, it's favored. All right, so that's how it works. And so I'm going to give you a little primer on outcomes from the gem, right? And so if this is a predator-prey model here. If you use a standard solver, you're going to get some squiggly lines. The solution for this a particular model is a set of abundances through time. So here's my prey abundances, here's my predator abundances through time. And if you run a Gillespie of some sort, you get something that looks like this. A whole bunch of squiggles. All of those squiggles are demographic stochasticity. And it's a lot because these are actually fairly small populations. And you see that some of them actually go extinct here. Right? So one of the things you can get out of the Gillespie evolutionary one or a non-evolutionary one is the probability of extinction. And that's nice. And then I'm going to calculate the median of all these different squiggly runs. But I have a hard time looking at all the squiggly runs. I just want you to know they're behind there. I'm going to show you something that looks more like this. Okay, so I'm going to give you these shaded areas to give you a sense of how much noise is going on. These ones are deviating a little bit from the ODE because of stochastic losses. So so I'm going to always show you some ODE version of my model. I'm going to show you my Gillespie outcome and then the range stuff. All right, so coming back to my DD logistic, right? What I want, I've broken down my parameters, and I want evolution to be part of my predictions, right? So we're trying to remind us what we're doing. We are trying to assess population outcomes. And if we're including evolution, right, that actually changes the outcomes. So in this case, in this simple example, I'm going to take my d-min parameter, I'm going to let it evolve using a gem. What I think is going to happen is that having a high likelihood of death is bad, that lowers your fitness. So we're going to evolve towards higher survivorship, which is going to lower this curve. And what you can see is happening is it changes the intersection point. So I'm expecting a population response. The population is going to grow basically because the carrying capacity is going to go up while evolution is happening. All right, so this is what I get. On the left-hand side, I've got mean trait here. The right-hand side, I got population abundance and time. This black line here is my trait. I started it out at 0.3, and sure enough, right, population evolves towards having a greater survivorship. Not very surprising. We started the population sizes here. The solid orange line is my gem. The dashed orange line is my ODE, so my ODE doesn't evolve. It goes to carrying capacity, which is the black line, black dashed line. But you can see there is a population response. Adaptation to a stressor, that's because adaptation to a stressor generally is going to increase the size of your population. Right? Because the stressor is reducing the size. So if you manage to deal with the stressor, you have a little bit of evolutionary rescue going on. That's the real reason why I think we should always be dealing with evolution and climate change, because if they are adapting, the population is responding by getting larger again. All right, so what I just said was you take a population model as sort of a mass balance constraint, and I use logistic as an example. Take apart the parameters to get at their mechanistic underpinnings. I only went one little bit here to GAR apart. Then I use a gem to assess real-time evolutionary effects on populations. All right, what did I just do in the landscape thing here is I was dealing with phenotypic differences. But you'll notice I didn't talk about cells or alleles or proteins at all. I took everything in there and I squished it down into one step. And that's because that's what quantitative genetics does, right? Quantitative genetics, using narrow sense heritability, assesses the statistical relationship between the phenotype of a parent and the phenotype of an offspring. I just know that there's associated with a certain amount of, with a certain slope, and that slope represents the amount of that trait that, or the proportion of the variance in that trait that is accountable by additive genetic variance. And I don't, a quantitative genetics mindset says I don't care how it comes about. I just know that it's happening, right? This is the kind of scenario where you're like, well, maybe I can start to do something down here. Right now, we're gonna try to get there a little bit more, but that's what we've done so far. All right, so remember this whole thing's about climate change, not just random evolution of parameters, right? So how do we deal with that? I want to use the same kind of approach, a slightly more complicated version, and actually kind of have some adaptation towards climate change. So where does climate change come in? Right, temperature is one of the big things, right? It's not just temperature, but where does temperature affect mechanisms or states in this landscape here? Anybody?
All over the place.
All over the place. Anybody second for all over the place? Second, third? Yeah, that's what I think. I think there is evidence that temperature per se is affecting each one of these mechanisms. And that's a bit of a problem. All right, how are we going to deal with that? We can't even deal with it even if we ignore the fact that there is a thing such as temperature, right? I mean, there is always a temperature at which everything is done, right? We most of the time just ignore that, right? But it's affecting everything. Fortunately, there are some ways in which we can actually see how temperature is affecting everything similarly. And one of those is a thermal performance curve, or TPC, right? TPCs are just a relationship between any kind of performance measure you can get and temperature, right? Can you measure at population level, individual level, cellular level, enzymes? They're typically unimodal. They usually have some sort of upper and lower limits beyond which things don't work so well. They usually have some sort of T-opt, and people like to characterize the breadth of the skewness of them and all sorts of things. And they definitely are more of an ectotherm kind of lens than an endotherm, because the body temperature of our ectotherms is a very strong function of environmental temperature. But these things do apply to endotherms. It's just that endotherms like to position themselves in some particular place. Right? But their enzymes and molecular reactions do the same thing as ectotherms. Right? But these things show up everywhere. They show up in enzymes. They show up in metabolic rate. They show up in sprint speeds. They show up in population growth rates. Right? So if it could be a useful way to simplify dealing with temperature across many levels. And here's an example. They're very popular right now as a lens of predicting effects of climate change and dealing with sort of community ecology. In this particular example, I love this one. This is a TPC of population growth rate for two Daphnia species. So population growth rate with temperature. In the New England lake, the native Daphnia is Daphnia culex, and this is their TPC here. And then there's an invading Daphnia, Daphnia lumholtzi. And it turns out the lake used to be at this temperature typically, and now it's warmed up a little bit. And the invader's TPC is shifted to the right. And so at that hotter new temperature, the invader actually has higher population growth rate than the native one, and is out-competing our native Daphnia. Right? So this is the kind of thing you can do when you're dealing with climate change. Okay, here's a change in my climate. What is the new performance of my organism? And it's another example of where community ecology and interactions between species is going to mediate or at least influence the effect of temperature on my performance. All right, so what I want to do next is just see, well, I wanted to also say that TPCs are sort of a way of visualizing the thermal niche of an organism, right? You might imagine if the world gets warmer or the world gets more variable in temperature, the TPC would change, right? That there is some sort of trait that determines what your TPC. In a hotter world, maybe your TPC should shift to the right, you'd evolve, right? You'd adapt that way. Or in a more variable world, maybe your TPC would spread out, right? So what I want to do next is actually just ask some questions using GEMS. Stealth is a trait to adapt to a changing climate. Right. This, given our Bmax of 2, right, this is happening by around 7 generations. So that's 7 time units. That's around 15 generations of time before it's moved over to 26. All right. Of course, climate change isn't just, switching our mean temperature from 23 to 26, right? It's doing all sorts of weird things, right? Temperature is already variable on daily, seasonal, multi-annual cycles. One of the things climate change, and probably one of the most concerning things about climate change, is the increase in the amplitude of cycles, extreme events, and possibly the periods at which cycles are occurring. So to really get at this question a little bit more, I decided to make my world in the model a sine function of time, or the temperature in the world in my model, a sine function of time, so that the temperature is moving at different kinds of amplitudes and periods. And then the question here is, okay, if temperature is varying, how does a population that's trying to adapt to a variable climate fare? Can it do that? Right? Can it track the environmental variation? And the answer is kind of. This is the same kind of layout as the last graph. Over here I have, you see that red line? It's a little bit faint. That's my temperature signal. So here I am at just at 23 again with stabilizing selection. And all I've done here is I've added some amplitude. So now it's moving from 23 to 24 to 22. And you can see that the population is in mean trait in blue is not too far behind the environment there. There's a little lag though over out at the ends. I'm not sure if everybody can see that, but so it's like, okay, not so bad. If I make the amplitude a little bit bigger, you can see the lag gets bigger. So it's definitely the case that it can be hard to keep track of a highly variable climate, even in a fairly simple adaptation kind of scenario like this. And then if I make the amplitude even bigger, there's even bigger lags. Well, okay, so I'm trying to predict population outcomes, right? That's my goal of climate change. Climate change is increasing the variances of my world, the temperature variance of my world. So what is my population doing in response to that? Well, it's kind of not doing that well, actually, right? So we crash and recover a little bit if it can adapt perfectly. If it's tracking okay, you can see that it's tracking okay here, but it's gone down, and it goes down on average a little bit and down on average a little bit more, right? So what you can see is that the average abundance is going down if there are lags in your ability to track temperature. But, I don't know if you guys see it yet, but probably the coolest thing as a modeler type that I saw in here was the fact that the populations are kind of erratic here, but they're absolutely cyclical over here, right? Something is happening that has generated cycles in my population that has no other mechanism for having cycles, right? It's a logistic type of thing, right? And I wondered why that was happening. So I'm going to draw some lines here from the peaks down. Here, it's a little messy looking, but just follow one of those black lines down, and where do you land? At the peak, this is a population that's doing good, right? At the peak, it's growing again. Where is that? Right there where the population mean trait is lined up with the environment. And then the population goes down during this little phase where the population cannot keep up with the environment. So it goes through this period of maladaptation. But because the temperature swings back around, it hits again a moment in time where you're really well adapted. And guess what? Population has a peak again there. And it keeps going through there. So what's happening is that one possible outcome of a population that is adapting to an increasingly variable world is that it's going to start to display sort of lagged adaptation cycles. This is something that I never thought about before, but this is one of the reasons why we do models, right? Because they can tell us things that you can't anticipate just by going outside much more. I don't know if this is true, if this could ever happen, right? But it's a testable prediction. And it's a way in which we can actually start to make predictions about populations, including evolution. All right, so I think I'm okay on time. I wanted to dig a little deeper, right, in terms of dissecting parameters a little bit more. Okay, so coming back to here, right, temperature is affecting everything. And one of the reasons why we think it affects everything is because it's, of its kinetic effects on molecular interactions, right? Reactions occur essentially through these sort of random contacts given a mass action sort of framework of things moving around in space, right? And that temperature increases the energetic state of reactants, increasing the rate at which they react. And we think that that happens very, very low down in the hierarchy. And as a result, people have used the Arrhenius equation to try to describe the effect of temperature on rates of, on reaction rates very widely now. It's the core temperature dependence used in metabolic theory of ecology. It's very widely used. It's analogous to Q10. Just has a different kind of framework. And this is how this equation works out. So we have R here is our reaction rate. A naught is our reaction rate potential. It's given by the availability of substrates, essentially, and mass action potential. This thing up here is your activation energy, which you guys have heard about undoubtedly, right? We got Boltzmann constant, and temperature in kelvins. Now, in this exponential term in the red box, this term here varies between 0 and 1. It's when temperature is at absolute 0, it's 0, and it goes up to 1. And that way we can understand why this thing is the proportion, the reaction rate potential. If temperature is hot enough for this to be 1, the reaction occurs at rate A naught. And then as it gets colder and colder, less and less of what's available reacts. All right, so we think of the Arrhenius, I think of the Arrhenius equation as approximately true, right? It is not actually mechanistically derived as far as I know. It's an approximation of another distribution. But it does describe chemical kinetics. Originally, it was used to describe the kinetics of reactions in test tubes. It's come from chemistry. But now we just sort of invoke it for biological systems, right? And I think that is why we're running, that is why I think the Arrhenius has a problem, right? Because it monotonically increases. But I just showed you GPCs, right? Pretty much all of the rates go up and then come back down. If our main equation to describe the temperature dependence of rates just goes up, we have a small problem, right? We need to bring it back down. And it's an interesting problem because we also think that kinetics are real, right? Like, actually, this is something that is real, and it is a mechanism that drives reaction rates. So what do we need? Well, a few years ago, a few of us in my lab and beyond had this question, or were trying to figure out what's missing. And so we went back to the beginning, right? So if you go back to beginning chemistry, biology, right, they will teach you that reactants turn into products through some sort of progression of a reaction. And reactants have some sort of energetic state. In order to get from here to here, you have to get over some sort of hurdle. Right? What that means is you have to get over this distance, which is represented as the activation energy, by increasing the energetic state of your reactants. Right? And as this gets higher, more and more things essentially spill over the top and react. And that's why the Arrhenius equation is actually really nice, right? Because it's literally, KT is literally the energetic state of our reactants. And you have to get the denominator to get higher and higher over the numerator for the reaction to proceed. So it's a kind of a nice thing, right? But again, this is what happens outside of bodies. Going back to beginning biology, what do they tell you? What do living organisms do to help their reactions progress? Enzymes, thank you. Right? We have enzymes, right? We take a reaction that is slow and we make it faster by lowering the activation energy. That is literally what the textbooks tell us happens. Okay, if that's true, what that means mathematically is our reaction that has some sort of reaction activation energy in a test tube, right, has some lower activation energy in a body because we contribute enzymes and we actually bring the activation energy down. So the observed activation energy in a biological organism is the difference between EB and EC. Okay with that? Yeah? Which means we can make a really simple substitution into the Arrhenius. This is really very, very simple looking, but for me it was like one of the biggest aha moments of my scientific career, because E sub C, how enzymes work, is temperature dependent. And so what that gives us is kinetics plus enzyme function. And it turns out enzyme function is unimodally related to temperature. There is an intermediate temperature at which enzymes work best. And that is characterized by something called free energy. And I'm not a thermodynamicist, so I skate very thinly with these terms, right? But essentially, free energy is the energy it would take to disrupt the function of an enzyme, right? To change its conformation so it doesn't work. And at its best temperature, right, it has a lot of free energy, which means it's going to work really well. And if it works really well, it lowers the activation energy a lot. And if it doesn't work very well because it's too hot or too cold, it doesn't do it very much. It makes it very hard. And so what this does is it transforms the Arrhenius that has this underlying physical kinetics into one that has this boost given by the enzyme performance. And it turns out we made an equation for this. I'm going to show you the equation. Sorry. You don't have to really look at the equation if you don't want to. It's super ugly, and I put it inside of a red box so you can ignore it. Right? But I wanted to point it out. All we've done is we've just gone back to the very basics and said, we think this is true. Here's the math behind it. We think this is also true, and here's the math behind it. And it turns out people who study protein thermodynamics have worked out an equation for for free energy. And it's derived. It seems to be pretty reasonable. I couldn't have done it. And this is what it looks like. Right, so this whole thing here is what we call the EAAR model, right? Don't look inside the red box if you don't have to, you don't want to. But if you want to, it's kind of cool. This thing here is enthalpy, which is essentially the robustness of your enzyme, like how fast it breaks down as you change temperature. This is a heat capacity, is actually sort of a temperature dependence term, and there's a melting temperature. And this is the true melting temperature, the point at which protein can't come back, right, after heating it up to that point. And this is just basically a curve here. And we stuck that in there. It's called the Bechtel-Schellmann equation. It goes back at least a few decades. And it's been used to describe enzyme performance across temperature for a lot of things. And it seems pretty good. And so now we have this, this expression here where, going back, you can actually get a T-opt out of here, right? So what I want, I haven't done it yet, right? These, I've broken out T-opt. I can break out T-opt into these things. I can break out T-opt into thermodynamic characteristics of proteins. And then I'm going to ask, is there an allele for that? I don't know, right? The cool thing about thermodynamics is everything has it, right? Everything has stability, right? Everything has enthalpy, right, relative to different temperatures. It's super general. The really not cool thing about it is predicting enthalpy of a protein from its structure. It seems to be, I'm not going to say impossible, but yeah, I don't know, it seems to me likely to be very protein specific. Right? Anyway, so now I can actually do the same kind of exercise and ask, well, you know, do these thermodynamic properties of my enzymes evolve in response to climate change? I haven't done that yet, but it's something we can do and then see what happens. But I took this model just to kind of show you again, we're crossing levels of organization with this. I took this model and I fit it to some metabolic rate data for an amphipod. Metabolic rate against temperature. This black line is the fit. Pink is the 95% model. So this model, using kinetics and enzyme performance, can fit metabolic rate really well. I put the model inside a biomechanical model of movement and fit it to some sprint speeds of geckos, and it can fit those really well. And we've also done population growth rate with the same thing. I'm going to show you that here. But first, I'm going to tell you why, about the data we fit it with. In my lab, in collaboration with Christy Montooth at UNL, we're doing a set of paired experiments trying to adapt two model organisms, in my lab, Paramecium, and Christy's lab, Drosophila, wild collected organisms, populations to hot and cold temperatures, so experimental evolution. I'm currently at close to 500 generations in my Paramecium, starting last September. Why I switched from birds to small things, right? Now we're going to do that with the bird, even barn owls, right? We're measuring thermal performance curves at multiple levels of organization, asking whether or not this mathematical model that describes metabolic rate drives all the other functions, right? You can't do anything without ATP. So if you can nail down metabolic rate, you might be able to nail down some of the other things. Right, so I just showed you the same, where we're at is we can do it for different things because we have different data sets. But here's the experiment. Basically get a bunch of paramecium from different ponds, mix them together, create an ancestral wild type, and then put them in petri dishes in my refrigerators all year. That's all, and feed them. Now, what I have is some data from April here. TPCs for population growth rate at different temperatures. This black dashed line is our ancestral TPC from the beginning. You can see all my population, all my replicate populations adapted to the lab. Right, that's what things do, which is why maybe lab strains are not good models for wild systems. Right, and it's kind of hard to tell if they're adapting, honestly. Right, over here at cold temperatures, these blue lines suggest that the cold ones are actually adapting to cold pretty well. But over here, I'm not sure that anybody's adapting to hot very well. And there's some difference going on up here. I'm not sure what it is. But okay, these are paramecium, a lot of plasticity. Presumably there was genetic variation in there. They're still alive. We're going to keep going, see if we can get them to adapt farther. But these lines, all of these fits are the here. So we've got a mechanistic equation describing temperature dependence that works for multiple levels of organization. The question is, can we make it work for any given system? And how can we transform one into the next? Going up. And can the parameters that describe that curve evolve? I don't know yet. Okay, so what I just am basically proposing, my hopeful proposal, right, is that you can take some place that you know to be true mathematically. You can make some part of that a function of some trait like T-opt. Emax is a function of T-opt. And that's something that's going on up here. And I can break that down now. My T-opt is some sort of function of my kinetics, baseline activation energy, EB, how well enzymes contribute to this reaction. That's maybe going on down here inside of interest. I don't know. I don't know if I put these in the right levels. The contribution of enzymes to the reaction is a function of the thermodynamics, the free energy of those enzymes. Free energy of the enzymes is a function of the thermodynamic properties of the proteins actually facilitating the reactions. We're talking probably basically ox-phos reactions here most of the time. I don't know, I know what these functions are, right, from Bechtel-Schellmann. But okay, the thermodynamic properties of my proteins that are running here are some function of some other proteins that go into it. I do not know what these functions are here, right? And these proteins are some other function of alleles. Down here, I don't know what any of those functions. Right, so what I think, we're at the point where we maybe can get down to here, right, and deal with rapid evolution in response to climate change and the population outcomes as a result, crossing at least a few scales, right? Maybe we can take one of Jonathan's models and slap it in here somewhere, and make some whole cell, efforts or something like that. I don't know. Right, but like I said at the beginning, I think this is a Herculean problem, right? Every single part of this problem is Herculean. So in total, it's really ambitious, but I think there are ways forward. That's my big message. And my suggestions about going forward is that you can make the math easier if you lock it down someplace where you can have sort of mass balance, energetic, some sort of constraint on the system. If you take each parameter not as a fact, but as an emergent property of things happening at lower levels, and keep dissecting them, you can get a long way. It's not that hard. It's not that hard any longer to allow for changes in allele frequencies to influence your population outcomes. The code on a gem is really, really easy. I'm not a great coder. I can do it. And it gives you, it can tell you how populations respond to different sort of scenarios of climate change and the gems. So we've got lots of people to thank. Dawn Foundation and PSF have been supporting our gem developments, and NSF is supporting our experimental evolution stuff, and a whole bunch of people who have contributed to GEMS and the lab work over the last few years. Anyway. Questions? Sorry, yeah.
So it seems to me that for a given enzyme that's evolving in response the shape of that curve.
Yeah,
And I'm
sure.
wondering whether you can imagine ways to predict for a given protein which one of those responses has the potential to occur.
Absolutely. Yeah, I can imagine, well, say in a gem, basically, you can have every single parameter and trait in the model evolving at one time. That's very, very easy to do. So, like, if you're invoking Bechtel-Schellman, as determining your enzyme function, you can allow the enthalpy, heat capacity, melting temperature to all be evolving at the same time. And it might turn out, and I think this is the thing that happens, a real thing, like, some things are really responsive, and other things just don't matter very much. And so we can do that, find out where are we likely to see some sort of change.
So, for one more question,
Yeah.
building on that. For the population of proteins that exist within a given organism, would you expect that which ones fall into the mostly optimal change versus shape change, that's predictable from the protein sequence because there's constraint exists, or is that a modelable thing, do you think?
It's so far beyond me, that part, I don't have any idea, right? I mean, and I'm not even sure how to conceive of, like, different proteins that are as a population of enzymes, right? Like, the curve that you get is always measured, like a protein function, protein stability curve is always measured on a population. And I don't know if that's like a whole bunch of different ones that just sort of add up, or, everybody's kind of the same. Like, is there the same level of individual variation among proteins as there are among individuals in a population? I don't know. Are there just a couple of types? Like, are there a handful of genotypes that have specific kinds of proteins that come out, specific versions of it? Are we dealing with a few discrete entities here? I don't know. Yeah. Valerie.
So I think what you said was models can tell us things that we can't anticipate, which I think was a good quote. And I think this morning we were talking more about models as predicting impacts, predicting changes of climate, effects of climate change. But it occurred to me some of the ways that you've been using the models are to to make predictions about mechanisms and to distinguish between different kinds of processes that might be driving patterns, which is another really important way to use the models and maybe to some extent overcome some of the challenges of validation that we have when we're trying to use models to predict the future.
Yeah, I mean, so like the one thing about this sort of evolution-induced cycles, right? Like that's a sort of qualitative type of thing.
Mhm.
Can we find cycles where we didn't find them before?
Yeah.
Right? Like you can go and look at, and I know there's actually changes in cycles of predator-prey things over time and across latitudes. There's some interesting patterns there. So that's this sort of model as a heuristic mechanism of understanding what kinds of things are going on out there.
Yeah.
Right? And this is not parameterized for any real system. But you could go back in and parameterize it for a real system and do both at the same time. Like do my cutthroat trout. What's the probability of extinction of my cutthroat trout as a result of increased variance in temperature?
So I'm wondering about the, say, universality of how temperature affects metabolism. So for instance, in depth theory, we assume that there is one thermal performance curve for all processes. And I know that in other models, in other applications, there is a thermal performance curve for growth, one for reproduction, one for feeding. So you essentially feed different ones for these different processes. So how do you see your model reconcile that? Or have you thought about, do we have one, so all enzymes function the same way, so all processes essentially follow one thermal performance curve, or do we need to separate those?
I hope they all follow mine. But
They don't.
I'm guessing they're not going to, right?
No, and in fact, Maxwell-Boltzmann distributions change with temperature. So the shape of the curve actually changes as you change temperature. And so you have both the substrate Maxwell-Boltzmann and the enzyme Maxwell-Boltzmann, which are two distinct things that are going to change with temperature differently. But I think what you did, I was really messed up by this whole thing here. I think what you did was you took a top-down approach.
I did.
So by doing that, it doesn't matter. So the mechanism is taken out of your model essentially. Other than you're inferring the thermal performance, but you're not saying why is the thermal performance different, which is what you're trying to get at, and that would involve those two maximal Boltzmann distributions changing shape, which would be a nightmare to try to model.
Yeah, well, so there are two things, right? One is I think you probably will have to allow for different types of TPCs for different kinds of processes, right? And for sure, where this is going to come up is things that involve cell membranes, right? Because cell membranes, function to stabilize things, and then they have sort of a little bit more severe kind of endpoints, sort of upper and lower boundary conditions. And so when then membranes really start to change, even if voxphos is still performing exactly the same way, the structure around them distributing stuff to mitochondria and pulling stuff away might change with temperature in a different way. So, but it depends on where you want to put that in. If you're modeling like the outcome of it, versus I want to actually explicitly incorporate a membrane at a cell level process versus say all my mitochondria at a within cell level thing. So it kind of depends on your goal. But that's an interesting point, Frank. I actually think we're not getting rid of mechanisms. We're just constraining what mechanisms are allowable given essentially mass balance of the system, right? So going down, there must be a thermodynamic characterization of the things driving this, right? It has to be there, right? And so we're just asking, is it, where is it? And then you can keep going, right? Looking for mechanisms, right? But every time you jump levels, right, you're invoking a mechanism. Right? Demography is a mechanism, right? Thermal performance curves are essentially a mechanism. Yeah. So, but as opposed to a bottom up where we're trying to identify everything down below and try to make them add up to the other thing, I am definitely saying this is the easiest place for me to mathematically lock this world down, and now I'm going to dig, to get to where I need to be, essentially. If that makes sense. Yeah, sorry.
Since you're running the laboratory inspection experiments, do you have any plans to do any GWAS studies later? I mean, you maybe have an opportunity to think about,
Yes, when I think about that,
functional alleles.
I say Christy, who's a proper geneticist, say Christy, here are my paramecium, good collaborator, take over from here, right? Yes, right. So we are aware that eventually, we could get to thousands and thousands of generations.
Yeah.
At some point, there are going to be some pretty radical differences among our populations, and it'd be nice to be able to dig deeper, and we'll know all these weird things. Right, we have measured the TPCs of metabolic rate.
It's incredible
Yeah.
span.
Yeah.
You can potentially integrate.
As long as they don't all die.
So you're getting metabolic rates on your individual paramecium?
We do them in batches of 30. But yes, pretty much.
And you're
Little
doing that
tiny
per
respirometers.
generation?
Over just a couple hours. So,
But each generation you've got a
No,
metabolic
no.
rate.
God,
No.
no. We're trying to line up our TPCs across levels at one moment in time. And we made a big effort on this in April and May, and haven't analyzed everything. We just have population growth ones are the easiest to analyze. So we have those. But we're going to do it again, hopefully when we get to around 1,000 generations. And so it's pretty straightforward. Protists actually breed a lot. It's really hard work to be a single-celled organism. Per cell, they are the biggest breeders. And so you can take 30 of them, stick them in a tiny little vial, measure oxygen respiration, oxygen consumption pretty quickly, and growth rates, cell expansion, stuff like that.
Thank you.
exactly where you're